Hey, biology. Welcome to notes on Punnet squares. Okay, so this is a new thing. Maybe you learned a little bit about this already from the readings and things like that. But we're going to be talking about Punnet squares. Punnet squares are a way for you to predict what's going to happen and what kind of traits you're going to get from when two parents make a baby. Okay, so basically, the idea here is we've already talked about genotypes, meaning the actual genes we get from our parents, one allele from our mother, one allele from our father to give us both alleles, which give us our traits. We get a genotype, and then our phenotype is what we express, what we see. Okay, and so what we're talking about is how can we predict what type of genotypes babies are going to get, and in results, what type of phenotypes are they going to get? Okay, now this guy named Gregor Mendel, he was the first person to kind of look at this stuff. And what's surprising here is that he spent a lot of time looking at this stuff, and he was around before we even discovered DNA. This is talking in the 1800s here. This is around the time of the American Civil War, really long time ago. Okay, this is before the invention of photography. It actually was just started at this point. I mean, photography had just started really, really long time ago. Radio wasn't around yet. It was just a crazy time. But he was looking at peas and kind of studying them. One thing he noticed is that there were different traits that peas had. He noticed some of them were round, some of them were wrinkled, and some of them were yellow, and some were green. And the flower colors had different color, violet and white and things like that. And he actually was able to predict how many pea plants, when they mated, were going to make round seeds, or how many pea plants were going to have yellow seeds. And he figured this out because he started using something called a Punnet square. Now, Punnet square is a kind of like a standard way of working out what the possible offspring of two parents will be. Now, remember, a parent can only give one allele at a time. So it's kind of like a probability. The idea here is we're looking at the four most probable combinations of alleles that a person would get. So the idea here being is that let's say we have a parent here and we have a parent here, okay? And this parent has a dominant and a recessive allele in their DNA, and this parent has a dominant and recessive allele in their DNA, okay? What we're saying here, this box, is saying, what if parent here gave their dominant, and what if this parent here gave their dominant, okay? This box right here is saying, what if this parent gave their recessive and this parent gave their dominant? And this one right here, what if they gave their dominant and gave a recessive? This right here is the probability, the 1 in 4 probability that both parents are going to give the recessive. Okay, so this is kind of a probability of what could possibly happen in an offspring. Okay, we conduct these two lines here. We usually make a grid. Okay, and we basically put the parents on either side. So we have one parent on top and one parent on the other side. Okay. One letter goes above each box, and it doesn't matter which goes on which side. The mom can go on top, the dad can go on top. It doesn't matter. We usually just call them it doesn't really make a difference. So up here, let's just say remember, this is our homozygous dominant on top, and this is our homozygous recessive on the side, just to give you little definitions there. Okay. Usually what we'll do is we'll just take them after we put them on the top, we'll just write them down. So we put the B's go down, okay, just like the arrow here, we put the B's down, distributed them, kind of those who are in algebra, we're distributing them down into the columns, and then we put the little b's over, okay? And what you notice is pretty interesting here is that when we have a homozygous dominant and a homozygous recessive, every offspring is 100% of the offspring are going to have heterozygous genotype. Very interesting. Okay, let's walk through some examples here. Okay, so we have some parents that are heterozygous. Okay, heterozygous means they've got one dominant and one recessive in their trait. Okay, and one dominant allele and one recessive allele. Okay, so the question is here, what percentage of the offspring will have violet flowers? Now, we know for them to have violet flowers, they have to have two of the little w's because it's a recessive trait. Recessive traits, remember, don't show themselves unless they're by themselves. So let's do the cross. Well, we have a big W, little w, and a big W, little w. Let's distribute here. Okay, so we've got a dominant, homozygous dominant, two heterozygous, and one homozygous recessive. So 1 out of the 4 is going to be violet. These are all going to be white because they all have a dominant allele inside of their Punnett square. Okay, let's see if I got that right. Answer 25%. Correct. So 1 out of 4, or 25%, will be violet. Okay, let's try another one. We got red hair is dominant over blonde hair. So big R is the capital R is for red hair is dominant over lowercase r is blonde hair. Okay, we're going to do a heterozygous redhead and a blonde. Heterozygous, remember, is going to be when we have 2 different alleles. Okay, and then blonde is going to be homozygous recessive. The only time you're ever going to have blonde hair is when we have 2 of the small r's. So here's our parents. One parent is heterozygous red, and one parent is blonde. Let's distribute out our letters. Big R's go down, little r goes down, little r goes across, little r goes across. Okay, so we've got 2 that are heterozygous and 2 that are homozygous recessive. What percentage of the offspring will have red hair? Well, we know that anything that has a big R is going to have red hair, okay? And anything that has 2 little r's is going to be blonde. So that's 2 out of 4, which is 50%. Okay, you can't see that right here. So my box right here is blocking it. But that's 50%, 50%. Okay, so let's continue on. That's 50%. Okay, let's try at least one more here. 1 or 2 more. In pea plants, tall pea plants are dominant over short pea plants, big T, little t. Construct a Punnett square for heterozygous tall pea plants and a short pea plant. Okay, very similar to what we just did. We're going to have heterozygous tall on top, and then homozygous recessive on the other side. Let's distribute. Question is, what is the percentage of the phenotypes? Okay, well, phenotypes. Oh no, what are we talking about phenotypes? Well, we've been talking about genotypes, which are the actual letters that are there. Here, let's talk about the phenotypes. What are we actually going to see? Well, over here, we're seeing those big T's right there. Those are dominant. So the dominant trait's going to show through. We're going to see 50% over here are going to be tall plants. And over here, we're going to see 50%. These are all recessive alleles, so these are all going to be short plants. So we're going to have 50% tall and 50% short. See that? What I want you to do today is I want you to go through, and if you're not confused, there's one more example after this. I want you to go through and I want you to practice this. I'm going to be attaching some practice problems for you to draw your own Punnett squares. It's going to be a worksheet. So I want you to either I want you to do the worksheet, print it out, or do it on a separate sheet of paper and send me a picture and put it on Google Classroom. So go ahead and work on this. This is a big part of genetics. This has been worked on for a long time, since I think my dad was in genetics back in the 60s. But this is just an important idea for us to figure out what the probability what traits will happen in our offspring. Okay, so go ahead and practice that for right now. Have a great day. Thank you so much for watching.